So after completing a doctorate in physiology from the University of California at Davis with John Horowitz and Tim Jones, Larry joined the laboratory of Vincente Honrubia at the Department of Surgery at UCLA School of Medicine, and he investigated primarily morphologic substrates of dynamic response heterogeneity among vestibular primary afferents. And, as you know, there's some fire with regular discharge and other with irregular, and some are very phasic in their responses. Some are more rate coding type of responses. And Larry and his team has played a large role in understanding how that works, in part through a collaboration with Michael Paulin in New Zealand, taking a different approach to probing how head movements code the role of spontaneous discharge and information transmission in sensory coding and sending signals to the CNS. Larry's laboratory focuses a great deal on ribbon synapses and vestibular hair cells, and it began with space flight investigations. So I had a beautiful picture of the space shuttle there to show, but we're missing that. And what he observed was that there was plasticity in synaptic ribbon densities after a 2-week exposure to microgravity environment on the International Space Station. Much of Larry's lab work is focused on the physiologic and cellular substrates of vestibular function and hypofunction, including those associated with behavioral agility and hypofunction induced by ototoxins and genetic modifications. And the research really provides a rich platform revealing novel insights into rehabilitation and neurocomputation. Now, I'm going to try to share the screen here one more time just to show you these beautiful images and pictures. So there's the space shuttle where that work was done. This is what Larry looks like routinely in the lab, I suppose, early in the morning, along with some friends and colleagues. This image is just outstanding. So that's the horizontal canal crista that out of Larry's lab, where he has labeled hair cells with tubulin. And you can see the calyces and type 1 hair cells labeled oncomodulin and calretinin. I think hopefully he's going to tell us a little bit about how these different features that we see here in immunohistochemistry relate to some of the physiology. Some of his work with Michael is really kind of interesting, and very interesting, looking at the way encoding is done in a little different way. This is a video that out of his lab. You're listening to a neuron changing discharge with head loss. I love the face playing, Larry.
Thanks, Rick.
Okay. I think hopefully Larry will tell us a little bit more about what we just saw when he began sharing his screen. And everyone, please join me in welcoming Larry for the kickoff seminar on this topic.
Thanks so much, Rick. All right, so let's go here. Okay, so I'm assuming that everybody can see this. So, Rick, thanks so much for the introduction. I really appreciate you moderating this session, and also thanks to Kathy Sung and the Education Committee for the invitation to lead off a distinguished group that will follow, that will include Kathy Cullen, Jay Rubenstein, and Chris Iluski in the forthcoming months. As the leadoff hitter, and since it is the fall, we will use an analogy to the Fall Classic. And as leadoff, it's important. I was asked to set the table and provide a little bit of background on peripheral vestibular neurobiology. And so, without further ado, this is the outline. Again, I'll provide a brief introduction, followed by I'll discuss a variety of models that we've used to try to understand some of the substrates, some of the cellular substrates of this modified function, but more importantly, what these modifications may mean for rehabilitation and neurocomputation. The models will include, as Rick introduced, a model of what happens when you change the operating environment under which the maculae work, and we'll focus on utricular synaptic plasticity that we found in a flight study several years ago. We've also been using the otoferlin knockout as a representative of a molecular synaptopathy. As most of you know, otoferlin is believed to be the principal calcium sensor in inner ear hair cells. And the knockout has some interesting ramifications in terms of the physiology of the epithelia. And what I'll discuss today are some of the downstream consequences. The other thing that's important about the otoferlin knockout is that it's an analog to the DFNB9 form of non-syndromic hearing loss. As many of the clinicians in the audience may know, the actual vestibular manifestations in the DFNB9 patients is not well understood. And perhaps some of these the knockouts may help us enhance that understanding. The title of my talk includes the word hypofunction, and at that time I thought I would stick to the models of reduced vestibular function. But in fact, I will talk about an animal that exhibits high locomotor agility, and it'll give you some insight as to what some of the adaptations may be in such a beast. And then finally, most of the lab work these days is focused on mild ototoxic lesions and how that manifests, what are the physiologic and cellular manifestations of hypofunction, again, induced by ototoxicity. So by way of introducing the topic and the vestibular periphery, this is a three-dimensional reconstruction from micro CT of a mouse, the bony labyrinth, the internal bony labyrinth, that can be relatively easily segmented. And so you see the horizontal superior and posterior canals. The vestibule is smack dab in the middle. The cochlea is underneath. And if we remove that, is revealed the sensory epithelia of the anterior crista, the horizontal crista, and the utricle. Let me get a laser pointer here. And this is the triad, so to speak, of these epithelia as stained with phalloidin to label the stereocilia of hair cells. And if we can take a section through the utricle, you might see something like this. This is a transmission electron micrograph of a mouse utricle. And I show this just to illustrate the two principal hair cell phenotypes. We're a creative group, and so therefore we call these two phenotypes type 1s and type 2s. And they are distinguished largely by the presence of the calyx. As Rick mentioned, the calyx is a specialization of the afferent dendrite, and in transmission electron micrographs, you can clearly identify the calyces that completely envelop the type 1 hair cell. Type 2 hair cells are not enveloped by the calyx, and so therefore they are also readily identified. The one thing I want to point out here is that these calyces are chock full of mitochondria, and you can see these mitochondria down here at the basal part of those calyces. The support cell layer is found down here. Now, again, if we take a little bit closer look at some of the cellular distinctions of these epithelia, we'll start with the utricle. And this represents a top-down view of a mouse utricle that has been immunostained with calretinin. Calretinin is a calcium binding protein that labels hair cells, primarily type 2 hair cells out here in the more perimeter region. And in the central region, it labels the calyces. And so therefore we consider this region, which is known as the striola, as the region that harbors the calretinin positive calyces. Certainly if we can identify the striola, then the regions outside of the striola are referred to as the extrastriola, and this has been further demarcated into the lateral extrastriola and the medial extrastriola. So the nice thing about the mouse utricle is that with a little bit of practice, you can clearly identify the topology and the orientation of this sensory epithelium. So this would be the lateral aspect of the the utricle, and therefore this is the lateral extrastriola. This is the medial aspect of the utricle, and therefore this would be the medial extrastriola, and rostral would be over here, and caudal is in this direction. So again, the striola is identified by the density of the high density of calretin positive calyces. The line of polarity reversal would run right along here. Okay, and very few of these calretin positive calyces are found on the lateral side of the line of polarity reversal. The other thing that distinguishes the striola is the enhanced expression of a low voltage activated potassium channel known as KCNQ4, as well as the presence of another calcium binding protein known as OCM expressed in both type 1 and type 2 hair cells. So if we go back to the phalloidin labeled sensory epithelia, the triad of the sensory epithelia, and we take a little bit closer look at the horizontal crista. Again, this is a horizontal crista from a mouse. It's distinguished from the two vertical cristae in that the horizontal crista in the mouse does not have the central region that is devoid of hair cells, known as the cruciate eminence. In this specimen that's been immunostained with beta-3 tubulin to label all afferent calyces and calretinin, you can clearly see that that most of the calyces in the central zone of the crista are calretin positive, but not all of them are. So here's a couple that are calretin negative. So in other words, the calretin labels most calyces within the central zone as well as the striola, but certainly not all of them. The voids that you see here that do not appear to show any cellular components, those are where likely the type 2 hair cells. So in other words, we can distinguish type 1s and type 2s by the presence of beta-3 tubulin and or calretin positive calyces. If we superimpose now oncomodulin, you can clearly see that this calcium binding protein, again, is concentrated or localized within the central zone. And again, you see a similar pattern within the striola. And again, you can see that within some of the calyces, you can peek through and you can see the oncomodulin positive hair cell poking through. But in other cases, you see oncomodulin positive hair cells that are not associated with the calyx, and of course these are type 2 hair cells. So in other words, the vestibular epithelia in mammals is certainly not a homogeneous epithelium, but exhibits functional and cellular specialized areas. We can identify and look a little bit closer at one of these afferent dendrites that is shown in this reconstruction. Again, the green represents calretin, the red is oncomodulin, and we can make the translucent, and you clearly see the hair cell inside. Okay, so in my lab, as well as others, including Rick's, various laboratories have made a direct assessment of labyrinthine output by interrogating the response dynamics of individual afferents. And in this video, what I'm showing is that the instantaneous discharge of a horizontal canal afferent that's modulated very robustly with a 1.6 hertz, approximately a 2 hertz rotational stimulus with peak velocities at plus or minus 15 degrees per second. And again, you can clearly see that the discharge rate of that afferent modulates, and it's pretty easy to see that the discharge modulation is a direct result of the change in head velocity. Now, the conventional way of analyzing these spike trains is to determine the peak and trough of the modulation, and in this case, the total modulation is about 8 spikes per second, or plus or minus 4 degrees per second, and then if that plus or minus 4 degrees per second is modulated by 15 degrees per second change in head velocity, gives you a sensitivity of about 0.3. And it's principally the peak of the discharge coincides with the peak of the stimulus. So the phase is approximately 0. And this has been a it's a linear stimulus, linear analytical methods that have been used for decades to illustrate the response dynamics in vestibular afferents. But if we take a little bit of a broader view, and if you look at the entire spike train, this is a 9-second epoch of a horizontal canal afferent. And you can see that most of throughout the entire epoch, some of the modulation that occurs here and here is about the same amplitude as that's what you see in the center. And you get a little bit more perspective by looking at the stimulus that was used to evoke this change in discharge. These magenta vertical lines are the occurrence of individual spikes, and so it's at this time base. It's the spikes are essentially fused, but certainly on my screen you can see that the higher density occurs essentially at the peak velocity of the turntable, and we can dissect this this epoch of discharge by looking at the per stimulus discharge from here to here. And again, the peak velocity has been ramped up in this particular case. And these vertical lines help you see that the peak discharge of the afferent occurs at essentially the peak stimulus there. And so it's pretty easy to see with these landmarks that this period of the discharge is a direct correlate to the stimulus that is shown here. But this also illustrates that there are stimulus-independent portions of the discharge, and it's this which we call spontaneous activity. But again, remember that spontaneous activity is really something that we can measure in the laboratory, but unless one holds their head completely still, which obviously rarely happens during normal behaviors, spontaneous activity is just a component of the spike train that is independent of the stimulus. Again, as Rick pointed out, there's a broad degree of heterogeneity among horizontal canal afferents. Again, we have a 3.2 hertz stimulus shown here. This is twice the frequency as that shown previously. So in my lab, what we do generally is when we double the frequency, we have the peak stimulus velocity. So it's essentially an isoacceleration stimulus. And again, you can clearly see that the modulation of discharge in the afferent is a direct manifestation of the modulation and peak velocity. And you can identify the periods of stimulus independence. But here's another afferent from the same animal, in which it's a little bit more tricky to distinguish without knowing where the rotation occurred, because the modulation of discharge in the stimulus independent regions is about the same amplitude as that during the per stimulus discharge. So again, we can in the laboratory we can clearly show that the discharge modulation is the peak discharge occurs at the peak stimulus. And again, we can use conventional methods to analyze the spike trains. The magenta shading shows the stimulus independent portions of the spike train. And this is a representation of a classic spontaneous discharge that was originally shown by Jay Goldberg and Cesar Fernandez in their classic papers from the 70s. These traces show the occurrence of the spikes, and you see that this one, the period in between spikes is very regular. And for this afferent, the period in between spikes is very irregular, and we characterize that with a parameter known as coefficient of variation. We put an asterisk by it because it's been normalized to a standard discharge rate of 15 spikes per second. So again, CV is just the standard deviation of the interspike interval divided by the mean interspike interval to give a normalized variation of spontaneous discharge. And again, this top afferent, the spontaneous discharge, was about 75 spikes per second, and the afferent in purple had a much higher CV and a slightly lower mean discharge rate. Okay, so what I've discussed thus far is some basic vestibular neurobiology that illustrates what some of the cellular components of the sensory epithelia are, and how that and the heterogeneity in response dynamics is exhibited by the spontaneous and evoked discharge among afferents. I also wanted to say a few words about how this neurotransmission occurs, at least quantal transmission occurs between hair cell and afferents. So this is examples from a series from the mouse utricle that was obtained through automated serial sectioning at 30 nanometers and using field emission scanning electron microscopy, so that we could actually reconstruct entire hair cells and synapses. A low power view is shown here on the left, and you can see that within the box there is a synaptic ribbon, which is the presynaptic structure that shepherds neurotransmitter vesicles to the active zone for release into the synaptic cleft. And again, this is the principal structure associated with quantal transmission. The video on the right, let's see, we can go back and we'll play that again. You can see that this is there's a serial with this series of field emission scanning images. You can be confident that we've captured the entire ribbon complex here, with the ribbon as well as the vesicles that surround it. This is a type 1 hair cell, and again, we know it's a type 1 because it's surrounded by the calyx. Again, and you can, I'll point out the mitochondria that are shown here. And so therefore, we know that this ribbon is a ribbon within a type 1 hair cell. It's a single ribbon structure, as opposed to others that we have found that are clusters. And so there's a broad heterogeneity in ribbon size and volume that we've found in the mouse utricle. We can also immunostain these for a little bit broader perspective on the distribution of ribbon densities and volumes. What I wanted to point out here, this is a single optical section in a confocal stack of a mouse crista. This is a mouse horizontal crista immunostained with oncomodulin, CTBP2, antibodies to CTBP2 that illuminate the synaptic ribbons. And again, the beta-3 tubulin labels the calyces and the parent axons. And you can see that some of these ribbons are within the type 1 hair cell, as shown in the EM series in the previous slide, but some are actually immediately outside. And it's actually, in actual fact, the number of ribbons that are actually outside of the calyx, and again projecting onto the calyx, there's actually the numbers actually rival those within the type 1 hair cell. And because we can identify them by virtue of being inside or outside the calyx, we can certainly identify those that are associated with type 1 hair cells, and those that are outside are associated with type 2 hair cells. I also want to point out the broad heterogeneity in size as represented by the CTBP2 immunolabeling. Now, these images were obtained using a Zeiss LSM 880 confocal microscope that was equipped with ARIScan. So this does represent an ARIScan image, which is Zeiss's rendition, at least at the time that we picked up this instrument. It was their rendition of super resolution microscopy. We now are applying methods of deconvolution, which actually helps reduce the distortion that is associated with all forms of light microscopy. And So this does give a bit of an inflated view of the volumes of these structures. And again, that's reduced with the application of deconvolution methods. Okay, so in summary of this brief introduction, we've been using immunohistochemical markers to measure their distributions, and we've spent quite a bit of time going to great pains to actually apply quantitative methods to actually characterize cells that are positively or negatively immunostained. And we do that primarily so that we can be sure, and we apply a strict criterion as to what we're calling positive and negative staining. And I provided a couple of references here that sort of will illustrate some of the methods that we use. What we hope to do is establish these as icons of sensory function and specialization. Again, through the use of, for example, markers for the central zone and calyx, and we can actually determine afferents that are projecting to particular areas of the epithelium by combining track tracing and immunohistochemical methods. By applying strict criterion to what we call a CDBP2 positive ribbon, we can also develop strategies for assessing changes associated with modified function, as we've done previously, and I'll refer to this in a few minutes. This is the reference to the spaceflight paper. And then we're beginning to use higher order analyses to assess functional modifications in the spike trains. And the goal here is to try to understand, with methods of inducing hypofunction, the goal is to identify physiologic changes that may be associated with the structural changes that we see through the immunohistochemical marking. So to start with, the first model of altered vestibular function would we embarked on a spaceflight study that was done a little more than 10 years ago, and the rationale is as follows. The utricle, again, as all of you know, are endowed with a accessory structure, the otolithic membrane that overlies the sensory epithelium. The otolithic membrane has a gel layer as well as an otoconial layer that imparts a high density material that makes this otolithic membrane sensitive to the ambient gravitational environment. And so during head tilt, the otolithic membrane would be displaced relative to the sensory epithelium, give rise to stereocilia displacement that would actually depolarize or hyperpolarize the hair cell. But in addition to head tilt, the otolithic membrane imparts the capability for encoding translational accelerations, as illustrated by the acceleration and deceleration in the cartoon. In the environment of the International Space Station, though, of microgravity, the head tilt responses would be absent, although the otolithic membrane still has mass, and by Newton's second law, it would actually, the otolithic membrane would be labile to displacement by virtue of translational head movements, just not because there's no or very little of an ambient gravitational vector. It loses the ability to encode head tilts. So when we started this, we thought we were going to use a method known as conical TEM tomography. And this provides a very high resolution, as shown here in this video. You can see individual vesicles, and with even a little bit higher resolution, you can actually see the lipid bilayers. And through manual segmentation or reconstruction, we were able to reconstruct parts of these ribbon clusters. But you can see that we're missing a large part of the ribbons because we were only using about 200 nanometer sections. And I know that Anna has used the facilities down at UCSD and using another form of TEM tomography to capture a little bit more of the volume. So knowing that we were missing a good part of the ribbons, we felt that we couldn't achieve our experimental goals. And so we went to an immunohistochemical method whereby we used immunohistochemical markers of, again, CDBP2 and SHANK1A to image different parts of the utricle. We imaged through the striola in 3 different regions, as well as the medial extrastriola. And again, we used immunohistochemical markers for CDBP2 and SHANK1A, and we counted the pairs and to give us a measure of ribbon density. We used the horizontal crista as a control that would be load independent. So it's believed that the changes in load that were encountered with residents in the International Space Station would not affect the function of the semicircular canal cristae. However, these sensory epithelia would be exposed to similar conditions as the maculae with respect to vibration and space radiation, etc. And what we found was that, again, when we assessed the synapse densities in the horizontal crista, basically the controls were not different than the microgravity exposed animals. Same thing with when we assessed the synapse density within the striola. Again, there was no difference between control and the microgravity. However, we found a 30% difference between controls and the microgravity in the medial extrastriola. So what this implied to us is that synapse density was labile to the ambient gravitational environment, and the decrease in density after unloading suggested a mechanism of that followed a learning rule of use it or lose it. And we would suggest that is a form of a synaptopathy. It does suggest that hair cells have the intrinsic capability to undergo synaptic plasticity. The fundamental mechanisms driving these changes is not expressly known, and we don't know yet whether synapse densities can be enhanced, which would be a nice if we could harness those capabilities. If they did occur, that would be a nice way to ameliorate synapse density in other forms of synaptopathy that are now known to exist. And we would suggest that this synaptic plasticity associated with spaceflight is a form of vestibular hypofunction. Recently, we had a chance to revisit this issue of microgravity induced changes in synapse density through an unmanned transport of a cohort of animals that were delivered to the International Space Station on the SpaceX Dragon. So this was a flight that launched in March, and it was spent 30 days on station and returned in the middle of April. This was a flight opportunity that was the mission was called MHU-8, and it was a collaboration with the Japanese space agency known as JAXA. So again, our previous flight results showed that utricular hair cell synapse density was dependent upon ambient gravitational load, and and the unique thing about this particular flight experiment was that the JAXA, or the Japanese Space Agency, built centrifuges aboard the International Space Station, provided the opportunity to investigate load superposition. And I think you could probably understand what the attractive nature of this experiment was. By virtue of placing the habitat cages at different radii on these centrifuges, they were able to achieve 4 levels of ambient gravitational load from 0G, in other words, controls that were placed in the same environment of the International Space Station, but just they were not exposed to the increased load due to centrifugation. So the loads were 0G to 1G, and it also included 0.3 and 0.6. And so the null hypothesis, or the fundamental hypothesis here, is that the 0G cohort plus the in the 3 other centrifugation conditions would yield synapse densities that were greater than that observed at 0G. And so this essentially would represent the intrinsic capabilities, or test the intrinsic capabilities of the utricle to undergo enhancements in synapse density if this condition is supported. And these specimens are currently in our processing queue. The next model of vestibular hypofunction is the otofurlan null mouse. And again, there's strong evidence that the otofurlan is the principal calcium sensor mediating vesicle fusion and transmitter exocytosis in most hair cells. And the otofurlan knockout, it's well known that these animals are essentially deaf. They have auditory threshold elevations of about 80 dB/SPL. And this is a representation of ABR, auditory brainstem response audiometry, shown in wild type, the otofurlan heterozygotes, which for all intents and purposes are identical to the wild types, and then the otofurlan null, illustrating that in our hands, these animals are essentially deaf. This is a copy of the vestibular evoked potential shown by Delon in 2009, showing that the otofurlan null, there is some vestibular function, although the amplitudes at a given stimulus magnitudes are diminished. And this is probably due to the retention of non-quantal transmission in these animals. So again, this evoked neurotransmitter release from type 1 and type 2 hair cells is severely compromised. Spiardi a couple of years ago showed that the compromise in evoked neurotransmitter release in type 2 hair cells is indeed the case. The original publication by Delon suggested that type 2s were spared, but they are in fact, by the time they reach adulthood, there is compromise in exocytosis in type 2s as well. This is our two recordings from afferents in my lab from the otofurlan nulls, showing that they exhibit a broad heterogeneity in spontaneous discharge, as one would expect, as is found in normal mice. And the distribution of oncomodulin positive hair cells in these epithelia is intact. So we found no difference between wild type and the otofurlan null with respect to the distribution of hair cells within the central zone, as illustrated by the same comparable distributions in the nulls compared to wild type. Like I say, in the central zone. However, we did see a dramatic impact on the postsynaptic expression of calretinin. The left horizontal crista shows the distribution of calretinin positive calyces in the wild type animal, and the right shows the otofurlan null, and you can see the number of calretinin positive calyces is severely compromised. It's dramatically attenuated. So this demonstrates that there's a postsynaptic dependence of dynamic activity of certainly this one factor of calretinin positive of calretinin positive calyces. Okay, so in the otofurlan mice, as a representative of a molecular synaptopathy, we found severe attenuation in stimulus evoked neurotransmitter exocytosis. And again, I emphasize the fact that this is stimulus evoked. We do believe that the spontaneous discharge is likely to be due to quantal neurotransmission, but of course we don't know that. And again, that may be called into question after the results shown at last year's ARO meeting, showing that the VGLUT3 knockout also shows spontaneous discharge. And again, the spontaneous discharge is preserved into adulthood in the otofurlan null mice. The idea of alternative synaptic tegments, to my knowledge, has not been specifically tested. So that is a potential explanation for why spontaneous discharge is preserved in the otofurlan null. But the fundamental result is that these data suggest the lability of postsynaptic components to presynaptic hypofunction, which will be very important to recognize, as many of you know that Decibel Therapeutics now has a dual AAV gene therapy to restore synaptic function in DFNB9 patients. So it would be interesting to know what the postsynaptic changes may be when otofurlan is restored in those patients, or in a mutant animal that they've also produced. And it's also important to know that despite the fact that we expect that non-quantal transmission is intact, that the non-quantum neurotransmission cannot rescue these postsynaptic deficits. So what about models of vestibular functional enhancement? And again, as we've shown, the crista central zone harbors cellular enhancements that support high fidelity coding of head kinematics. And we know that the central zone, crista central zone, harbors the highest afferents exhibiting the highest sensitivity and phase lead. And phase leads in those afferents in response to changes in head kinematics. So the question is do animals associated with highly agile behaviors exhibit cellular enhancements in the central zone? And this is not a unique question to us. Anna Lisakowski's lab showed a number of years ago in looking at the vestibular epithelial models from mouse through the ground squirrel, again, representing a broad heterogeneity in behavioral locomotor agility, that many of the central zones exhibited similar features among those animals. So it appears as though there are no enhancements shown in rodents. So what we did is we actually looked at another highly agile animal. This is Rosetus aegypticus, or the Egyptian fruit bat. And in these animals, we did an examination of the relative changes in head kinematics relative to body kinematics. The idea here is that the blue or the cyan lines, the cyan traces represent the body acceleration in Z that is parallel to Earth gravity. So the body is modulating acceleration with each wing beat frequency shown in cyan. The red trace represents the GZ, or the acceleration, vertical acceleration of the head. And you can see through most of this flight epoch, this short flight epoch, that there's evidence of head stabilization as shown here. In other words, if the head were rigidly coupled to the body, then you would expect the same excursions. But since it does not show the same excursions and acceleration, it means that the head is not rigidly coupled to the body. And we believe that this is evidence of a behavioral form of head stabilization. Shown down below are the identification of the sonar emissions that these animals exhibited during flight. And we've indicated what we did is these animals emit sonar pulses in doublets. And the first doublet is identified and shown here as the blue triangles above. And what this illustrates is that the probability of a sonar emission occurring during head accelerations greater than 1G is about 70%. And again, we believe that this is evidence of a stabilization of acoustic gaze. So we looked at the expression of some of these fundamental markers within the epithelia of Rosetus and compared that to mouse. And whereas the mouse, the fraction of calretin positive calyces relative to all calyces is about 70%. Okay, and Rosetus, though, that fraction increased to well over 80%. So we believe that we would advocate that this is a representation of a natural adaptation to these highly agile behaviors. Again, what is shown here is that the wing beat frequency is about 8 hertz, which is higher than what a terrestrial rodent would experience during normal ambulation. So again, in summary, Rosetus exhibited head stabilization behaviors to counteract the large excursions in body acceleration during 8 hertz wing beat frequencies. These animals exhibited an association of their sonar emissions, and it suggested head stabilization is advantageous to establishing acoustic gaze stabilization. And we also found that there was an enhanced fraction of calretin positive calyces in the crista central zone, and it appears to represent an adaptive modification to the demands of agile behavior. I'll move through the final portion of this seminar relatively quickly, because I see we're already at 50 minutes past the hour. So this is a representation of head and eye movements in a patient that experienced bilateral vestibulopathy induced by intratympanic gentamicin. And you can see that under normal conditions, in a non-pathologic condition, that the eye trace would match the head trace to give a gain of about 1. But instead, in this condition, the slow eye movement associated with this head movement, and again, this is using the vestibular head impulse test, is about half the amplitude of the head movement, amplitude of head acceleration. And it's also associated with overt and covert saccades that follow the slow eye movement. And this is a signature response of vestibular hypofunction. So in trying to develop an animal model of this, what we decided to do is eliminate the ambiguity of systemic administration of gentamicin by infusing the gentamicin directly into the perilymph and then waiting various periods of time. And in this first experiment, we used a total of 1 microgram gentamicin, again, infused directly into the superior canal, into the perilymph of the superior semicircular canal. This is the results after 6 months. These traces up on the upper left, the top traces show the deterministic head movement that is shown in an anesthetized preparation and recording from individual afferents from the superior and horizontal canal. And I showed these 2 traces just to illustrate that even though the amplitude of this modulation is very low, the coherence actually ends up being quite high, because these animals have a very low CV. In other words, the component of the discharge that is independent of the stimulus these are very regular afferents, meaning the noise component is very low, which drives up the coherence, again, despite the fact that the amplitude of the response is very low. The next traces represent 10 consecutive afferents in that preparation, in a preparation, again, that had received 1 microgram gentamicin. And you can see that in all these 10 afferents, the coherence is quite low, indicating that there is no evoked responses in these afferents, despite the fact that the spontaneous discharge is preserved. These next 2 micrographs illustrate what the morphology of the lesion looks like. Again, this low power view of this chinchilla crista shows the distribution of calretin positive calyces, and it's shown with the tubulin positive calyces shown in this high power view. Again, we're looking straight down upon the center part of the central zone, and this is what the lesion crista looks like. All the calyces appear to be gone, but the parent axons are still there. This is another representation. We typically, in that experiment, we use the contralateral ear as a control, and this is a reconstruction of the hair cells and calretin positive afferent calyces. And this is what the specimen looks like under conditions of the 1 microgram gentamicin, again, 6 months after infusion. We also observed a modest hair cell loss of 20 to 30%. Okay, so in this first experiment, the partial lesion induced by 1 microgram gentamicin illustrated the calyces are very sensitive to low-dose gentamicin, though the parent axons and most of the hair cells are actually preserved. Calyceal lesions are not the result of afferent apoptosis. Again, there was no change in the number of afferents. We observed a preservation of spontaneous discharge in the absence of evoked discharge indicative of a presynaptic lesion, and that this pathotype is similar to the functional deficits observed in the otoferlin null mouse. And again, we did not assess the presence of otoferlin in those gentamicin exposed preparations. In summary, then, it appears as though the clinical vestibular hypofunction, as shown in that first slide, was better modeled with experimental doses that were less than 1 microgram gentamicin. because the 1 microgram gentamicin, you would not expect any eye movement simply because we could not record any evoked discharge when directly assessing the afferents with single unit electrophysiology. So how might we explain the changes in the epithelia that may be associated with a hypofunction condition as opposed to complete paralysis? Again, this is the slide I showed earlier showing the discharge heterogeneity. Basically, the challenge to sensory systems is the proposition of solving Bayes' rule. In the laboratory, we can clearly assess the probability of a spike occurring because we know what the head is doing. We apply deterministic stimuli, and then we measure the spike train that follows or is associated with us change in head kinematics. The central vestibular networks, though, under natural conditions, is quite a different situation because the brain is charged with the task of the information it gets is from the spike train coming from the afferents, and it has to assess the head kinematic state that is based upon measurements coming from the ear. And I provide a couple of asterisks that I'll come back to in just a couple of seconds. Part of the ambiguity here is that even in the absence of the head kinematics, there's a component of the spike train that is stimulus independent. In other words, the probability of a spike occurring in the absence of head kinematics is not 0. So we characterize that component that is not linearly associated with the stimulus, we're going to call for in the present context as noise. But in fact, it's not noise as something that is to be discarded. It's something that the brain has to deal with. The inference here is that vestibular afferent spike trains are encumbered with measures of uncertainty with respect to individual spikes. Therefore it's representing an uncertainty with respect to determining what the state of head kinematics is. The asterisk here refer to the fact that even though I've characterized this challenge as determining head kinematics and the only information is coming from the spikes coming from the vestibular epithelia, in fact, there is likely to be some state dependence or predictive sensing, as has been advocated by Kathy Cullen and work from her lab, and we'll hear from about that in a couple of weeks. So the question is there a measure of the entire spike train reflecting the degree to which it represents the stimulus? I would advocate that this is characterized by this measure of coherence or stimulus response coherence. Coherence is a spectral measure of the entire spike train, and it represents the degree to which the response is linearly related to the stimulus. It's essentially a representation of the signal-to-noise ratio, and again, where noise is a component of the spike train that is unrelated to the stimulus. To illustrate these measures of coherence, I show a couple of traces of afferents with relatively high coefficient of variations, although the sensitivity measures are quite different, and that manifests into quite different measures of coherence. Okay, the coherence is quite high in the upper trace, but much lower in the lower trace. These are, again, representative examples of spike trains that exhibit low coefficient of variation. And again, there's also heterogeneity in the measures of coherence. In this plot, I'm showing you untreated or normal afferents, illustrating showing coherence as for afferents exhibiting different measures of coefficient of variation, where the size of the symbol is an analog of afferent response sensitivity to a 1.6 hertz stimulus. If we separate these two just arbitrarily at 0.1 and do a little non-parametric statistical analysis, you can see that there's a big difference in the sensitivity measures, but no difference in coherence measures among in afferents with low CVs compared to those with higher CVs. We take a different look at this, and we're looking at the distribution of sensitivity relative to the coefficient of variation, and I've included here afferents that were recorded from animals dosed with lower doses of gentamicin of 0.5 and 0.75. Remember that the 1 microgram gentamicin, there were no responses, no evoked responses. And in this case, we do a resampling statistical strategy using a measure that is known as Kullback-Leibler divergence, which is just a way to compare distributions. And basically, this tells you that there's no difference in the distributions of sensitivity between animals that receive these doses of gentamicin compared to untreated. In other words, the resampling paradigm allows you to ask the question, what is the likelihood that I could have derived the sensitivity measures from these low-dose gentamicin out of the entire population? And the answer to that is the probability is relatively high. However, if we now plot coherence versus CV, and again, you see these afferents that were treated with low-dose gentamicin among untreated afferents, you see that the likelihood that the coherence measures of the treated afferents could have been derived from the entire population is actually pretty low. So in other words, this indicates that the coherence measures of the treated afferents is lower than untreated. So it appears as though that gentamicin, whereas it doesn't have an impact on a conventional measure of response dynamics, it does impact this measure of signal-to-noise ratio. So again, in summary, the gentamicin results in attenuated head impulse response gain. The present data that I showed you from our laboratory shows that low-dose gentamicin reduces afferent coherence, and this implies that the vestibular neurocomputation relies on the entire spike train. In other words, if this response was driven primarily by or associated with conventional measures of afferent response dynamics, in the gentamicin case, you wouldn't necessarily expect there to be this change. However, because we don't see the change in gain or phase in our low dose measures. However, there is a compromise in coherence. So it appears as though this result that was obtained in patients may be associated with the diminished coherence. And this has a very different implication for neurocomputation, similar to some of the models that have been advocated by Faisal Karmali. This also indicates that we can perhaps design rehabilitation strategies to, rather than try to bump the responses, to actually diminish the noise component of the spike train. How would you do that? By making them more regular. Just very briefly, this is what the morphology of the cristae, how they appear in these low-dose conditions. And I also want to indicate that a big part of our work now is associated with synaptic assessments. This is a immunohistochemical staining from a cross section, a 25 micron cross section showing CDBP2. We're using Homer now as a postsynaptic marker. And the white illustrates that CDBP2, and the calretinin and the calyces indicates that this is from the central zone. So in summary, I think we can conclude that the modest ototoxic lesions result in diminished afferent spike train coherence and their response fidelity relative to head kinematics. Though a presynaptic component to hypofunction is likely, ototoxic hypofunction also impacts afferent dendrites. We've made indirect reference to diminished VOR gains, suggesting that central neurocomputation does not rely on conventional parameters of afferent dynamics, and we're going to, in the process of developing or reinvigorating our VOR measures in my lab so that we can make these associations much more direct. And again, perhaps this provides evidence that there are different strategies to rehabilitation that might include a reduction in spike train noise. So again, in conclusion, these are I wanted to acknowledge the individuals that were principally responsible for doing the work. Dave Saltemeyer was a postdoc in the lab. Amy Tan and Terry Prince were graduate students. that were responsible for the BAT and the otoferlin null work. Kevin Wright, Cassandra, and Tyler. Because Kevin just left the lab to start a graduate program at Pitt. Cassandra and Tyler are currently in the lab, and Tyler will be processing the new flight specimens from the Dragon flight. Jackson was principally involved in the analyses of the bat head and body kinematics, and Walter Metzner, Ivan Lopez are intramural collaborators, and I've been collaborating with Ivan and Felix for some time. Shout out to Mike Pollan. Mike King is a consultant on our funding to help us do the VOR correctly. Jeff Lichtman and Richard Shalak are at Harvard, and they were responsible for, and their collaboration was key to getting our field emission electron microscopy data. And of course, Cynthia Moss and Angie Salas were at Cindy still is at Hopkins. Angie is now at University of Illinois, starting her first faculty position, and they were involved in the flight behavioral work, the Rosetta's flight behavioral work. And these are the funding opportunities, the funding agencies, and I greatly appreciate your attention. Thank you very much.
Well, let's thank Larry. That was wonderful, Larry.
Thank you.
We are just a little bit over time. So I think we'll have maybe 5 to 10 minutes at the most for the question session here. And Sarush, if you're still online, you had one in the chat, two in the chat that are interesting. I think you could open your mic if you're still online here, Sarush.
Sure. Hi, Larry. Thank you very much. It was a really good talk. Very interesting. Very wide range of material. I have a question about the space flights. I would ask that first.
Yeah.
So when the animals are traveling through space and coming back, there's a huge amount of acceleration there, both times. Do you think that would have any effect? It's like a huge noise for cochlea. You would expect to get some damage to the vestibular end organs.
So the animals are not. There's certainly no evidence that there's overt acoustic trauma. Okay. This is an age-old question, Sarush, and every flight geek has to deal with this. And we're concerned more with, there's two things that we're concerned more with. One is the vibration. Okay, obviously there's you're absolutely correct that the acceleration excursions during not only the travel to space station, but on recovery, you have to recover all that energy on return to Earth. So certainly that will be that's a consideration in the first flight studies. And again, we weren't the first one to look at the vestibular epithelia, as you know. I mean, Muriel Ross started it almost 40 years ago. So in those conditions, there is no real control for that, and that's just an anomaly we have to deal with. In the Dragon studies, though, we will have 4 different cohorts, some of which were exposed to the centrifugation, and some of which are not exposed to centrifugation. And so that there will be controls where we can look at this a little bit more carefully. Also, if there was some general phenomenon such as acoustic trauma to the vestibular epithelia or vibration, you might also expect that to affect the non-load dependent epithelia, such as the Christi. And again, you saw that the Christi are unaffected. So we do believe that the changes in synapse density in the utricle were specifically due to the changes in ambient load.
Mm-hmm. Okay, thank you.
Yeah, I don't see any hands at the moment. Larry, let me ask you, so a lot of the single units you showed us with coherence were like 10 degrees per second, 15 degrees per second, yet the head thrust is 200 degrees per second, over 150 millisecond duration, which one would expect. It's almost surprising in some ways, based on single units, that the head thrust impulse test tracks head velocity so well, when in fact the afferents don't look like that in population. So can you
Right.
comment on that?
Yeah, so we put it's not just in the head thrust, Rick. I mean, this is a problem. We put a accelerometer on the head of a chinchilla, and we let him walk around, and we measured the head acceleration just in casual locomotion. And even in our complex stimuli, which was which was shown in that the video that you actually showed, the head kinematic space is in that complex stimulus. If we can represent it like this is the state space that of head acceleration and velocity. Yet during just casual locomotion, it incorporates a much larger space of head acceleration and velocity. And so the question is how well does, when we design stimuli in the laboratory, because obviously if we try to mimic the thousands of degrees per second squared of acceleration, or even much higher velocities, the likelihood of holding units is obviously much lower. And so, yes, we can couch it into as a linear system, that it would be representative. So, we just don't. I don't know how that would impact. Clearly, at these higher accelerations and velocities, the high accelerations are very small amplitude, right? But higher frequency. So clearly that would involve perhaps much more non-quantum. But the fact of the matter is that the direct correlate between a behavioral head movement, again, when an animal is running, like a chinchilla is a very agile animal. They're running around the cage, they're leaping. Their natural environment is the mountains of the Andes. We'd like to think that the measurements we make in the lab are analogous, but there's no direct proof of that.
If anybody has any questions, please raise your hand. But I have another one, Larry. I thought it was very interesting about the type 2 synapses loss in zero gravity.
Yeah.
And I'm kind of curious if you had animals of different ages because of the differences in, type 1 versus type 2.
Yeah. So we don't. So as you know, NASA, they prescribe a C57 Black 6 mouse of specific ages. All the animals are age matched. So we don't have different ages of that. They were 8 weeks. So more importantly, as you know, Gabe Korfas's lab has shown there's an age-related synaptopathy. We're certainly not at that point. So we don't think that that any synaptopathic, certainly between the controls and the flight animals, they're the same age. They're at harvest, they would have been 10 weeks old from the SDS-131 flight. These animals that were just flown are a comparable age.
That's really interesting too, that the loss that you're seeing in that situation seems to be like the opposite of what you would get with vibration-induced loss or noise-induced loss.
Yeah, yeah. So as you know, Muriel Ross reported an increase in synapse density and primarily in type 2s, very little in type 1. But I got to tell you that that's where the integration of the EM and the immunohistochemistry is helpful. From the immunohistochemistry, so for example, Muriel reported mean synapse counts in rats from type 1s to be about 2 to 3 ribbons per hair cell. And she reconstructed individual hair cells, at least in some of her specimens. Anna and Jay reported much higher numbers in type 1s. And our immunohistochemical results also illustrate that the number of ribbons per hair cell are much greater than what Muriel reported. So I don't know how I can't speak to, how Muriel got her counts, and how it reconciles here, but we feel with immunohistochemistry, it's a different approach to be sure, but we're also sampling a much larger area.
Okay, and
mean-
there's a question in the chat from Dr. McPherson regarding the fruit bat head stabilization data. Do we know if that's vestibular?
We don't specifically know, because that could clearly be a component of, as Kathy might argue, is there's other inputs that could be impacting, and we would certainly advocate that if you take a Bayesian approach to central processing, that these animals are going to use all the information available. So in the case of this locomotion from spinal pattern generators, so I don't know whether the adaptation seen in the epithelia are direct manifestation of the agility, or that the head stabilization is specifically vestibular. We'd like to think so, but we have no evidence of that.
I see Anna has her hand up. Anna, can you unmute?
Yeah. I think to Larry's point about Muriel's data, I think part of the issue was that she was using albino rats, and
Fair enough.
that with normal rats there are slightly more ribbons, but still not as many as in mouse and chinchilla, because we've been doing the comparison. So I think that was part of the issue.
Thanks.
Yeah, okay, fair enough. I don't recall that they were albino, but you do. So
Yeah.
yeah. So
Spargdali, I think.
I think it's we can just say that there's a potential sampling error, potentially something in we know that the albino inner ears are different. So
Rada, could you unmute and ask your question? I see you have one in chat there.
Hi, Larry. Lovely
Hi, Rada.
talk.
Thank you.
I'm curious about the otoferlin knockout mice. As you know, we've been sort of interested in some of the results we saw in the VGLUT3 knockout mice during ARO. I'm trying to get a feel for, do you think the otoferlin knockout is having some other effects on hair cell physiology that might be impacting the transmission of information from hair cell to neuron that the VGLUT3 knockout might not be? So having an effect, for example, on the non-quantal component?
Yeah, there's no evidence of that. We have no evidence of that. I don't know. We do know that from Dulon's work from 13 years ago, 14 years ago, that there's no change in capacitance. And the spontaneous discharge, so they also recorded. So again, Dulon was used very young animals, I think younger than P11. And that's where he distinguished this impact of otoferlin on type 1s versus type 2s. The later study from in collaboration with Walter Mercati, they actually looked at capacitance measures in adults. And so that the deficit, again, the absence of changes in capacitance in type 2s was also found in adults. Now, whether there's any other changes in that may impact non-quantal, I don't know that we know that at all.
Okay. We're about out of time, but I see Sarush has his hand up. Why don't we take one last question? If you have questions
I just
beyond
want to say that with the otoferlin knockouts, you showed that the calyces were kind of damaged. Am I right in your labeling?
No,
You show fewer
yes,
calyces.
no, it's sorry. What I didn't. So we actually there's no change in. So we also use tubulin. So there's no change generally in the calyx. It's just calretinin expression.
So there
it's
is a
the
change.
fraction.
I'm not sure we see that in VGLUT knockout, so that can affect the responses.
Yeah, potentially. I'm not sure that we know what calretinin is doing postsynaptic.
Right. But that's a
Yeah, but I think the important thing here is as we transition into, I mean, in my view, this AAV will be one of the first probably success stories of genetic rehabilitation in the DFNB9 patients. And again, I think it's important to recognize that in the vestibular epithelia, there is likely to be alterations postsynaptically.
Right.
So it's not just a linear removal of otoferlin. There's other impacts.
Yeah. Well, Larry, thanks for the great talk. I think we're about out of time here. Others that have questions, please feel free to contact Larry directly. And I really enjoyed a beautiful talk, Larry. Thanks.
Thank you very much, Rick. Appreciate it.
Thank you, Larry.